Out of this because they are key points that you will discuss in your lecture. And if you have questions, you can come back and ask me about them. The other thing is that I want to do protein synthesis as well. And that's kind of a long presentation. And that will probably be next week, next Monday. So we're doing DNA today and proteins, protein synthesis next week. So you may look over it so it doesn't sound so foreign. But I'm going over it now to kind of help you out when you come across it in lecture. So this is one slide, but it's kind of a big story here. And the person that did this research, his last name is Griffith, and pretty much known for what this slide says, the transformation experiment. So, can anybody tell me what transformation means? Transformation. What is transformation?
Change states.
You're going to change, that's true, somehow. And that's what it means all over the world. And genetics, it has a kind of a different meaning. And let me try to give you both of them. In medicine, when you hear the term transformation, they mean going from non-cancer to cancer.
Can you repeat yourself?
Yes, I sure will. In medicine, when you hear the term transformation, they actually use it to mean going from non-cancer to cancer. And that's the definition that started very, very early when they were trying to understand what causes cancer. And so they said a cell can be transformed, meaning that it's going to go from normal to cancer. And to this day, they use that definition. So a transformed cell in medicine, in the medical field, means that it's cancer. Griffith was not using it that way. He was using the true definition of transformation, which means that a cell will receive foreign DNA, and that foreign DNA is expressed. So that's the strict definition for transformation. One cell will receive foreign DNA, meaning from some other source, and that DNA enters the cell and it is expressed. And that's the definition of transformation for you as genetic students, but it's good to be aware of both of them. Griffith did his experiment in 1928, so they did not know what DNA was in 1928. Had no idea. Matter of fact, this whole presentation is built on what is the genetic material. And this is a classic demonstration to prove what is the genetic material. And basically he worked with an organism called pneumococci. And can you think of what disease pneumococci will cause? Pneumonia. Pneumococci. So pneumococcus, pneumococci. So pneumococci can cause the pneumonia. But here he has two different kinds of pneumococcus. Two different kinds of pneumococcus. One is a smooth, and you can see the S's by certain of the words, the letters, and one is rough. And is that clearer? I wear glasses, so I'm asking you. Is that pretty clear for you? Okay. So one was called smooth, and the other one is called rough. One is virulent, the other is non-virulent. So what does the word virulent mean? Virulent means it causes the disease. It's going to cause an illness. Matter of fact, I could say it's pathogenic, virulent. And non-virulent will not cause a disease. And the difference between them two, as this slide indicates, is that One has a polysaccharide coat, the other one does not have a polysaccharide coat. So the one with the polysaccharide coat they call smooth, and the one without the polysaccharide coat they call rough. So obviously you can see the trend of thought is going to be the polysaccharide coat is causing the disease. So the smooth one is virulent and the rough is non-virulent. And so the opinion of many would say what's obviously seen on the surface is that one has a coat, the other one doesn't. So maybe that's the cause of the virulence. But Griffith was asking what causes a cell to change. Now I gave you the definition of transformation, but keeping in mind that Griffith did not know what DNA was. It didn't have a name in 1928. Surely polysaccharides were around, proteins, they knew what proteins are, and they understood what lipids are. But they had not discovered the this molecule called DNA. So many of these experiments we're talking about today that I'm going to kind of gloss over is asking the question, what is the genetic material? And of course that's easy for us now. We know the genetic material is DNA. But back in 1928, 1940s, 1950s, they were still thinking that the genetic material could be a protein or could be protein. And that's what this whole discussion was about. After which we will do a short lab where we will isolate DNA. So moving right along, back with Griffith, he was working with two different kinds of pneumococcus. If he injected the smooth, the mouse would die. If he injected the rough, then the mouse would live. But what complicates this even more so, there were 3 different types of smooth and 3 different types of rough. But all of the rough are non-virulent, all of the smooths are virulent. So whenever you inject a smooth into the mouse would die. And of course he inject the rough, it would not die. And there are 3 types of virulent, type 1, type 2, type 3. And there are 3 types of rough, type 1, type 2, type 3. But what's interesting is a smooth cannot mutate to become a rough. And a rough cannot mutate to become a smooth. And I say that because obviously there are a number of genes that's involved in making the virulent smooth, and a number of genes involved in making the rough rough. So, you know, a mutation in a single gene can't cause it to become smooth. So obviously there are a number of genes involved here. So going through with the experiment, if he killed the smooth, if he killed the smooth by heating it, in other words, if we increase our temperature to like 109, what will happen to us? So I don't have to heat you up to boil it. I can just raise your body temperature up to where everything starts to denature and become inactive. You will not survive. So that's the kind of heat I'm talking about. So he will heat up the types 3 smooth and inject it into the mouse, and nothing happened. The mouse lived. Obviously, heating it up killed the bacteria, so nothing happened. Now, this is the part of the experiment that kind of is occurred. If he mixed the heat-killed smooth with the live rough and injected that group into the mouse died. And when he does an autopsy, he gets live type 3S.